I swear I didn't do that. I can see it right here. Oh, okay. I could just give the talk, and you would never know if I was telling the truth or not, and then you'd walk away and just believe it all. Okay, so we do single cell analysis because, again, we're interested in heterogeneity. So do any of you guys use this cell profiler? It's freeware. It's great. It can be modified using MATLAB, so that's what we did for our particular data set. So we wanted to isolate the cytoplasm and exclude the nuclei of all the cells, and we wanted to only keep cells within a given diameter range because we wanted to exclude cells that were not tumor cells. And from these measurements, we have 8 variables, and we extract these for each cell. And in order to quantify the heterogeneity of the sample, we actually plot histograms of the number of cells for each of these variables. And that's what you see in blue. That's a histogram. And we fit this histogram to 1, 2, or 3 Gaussians, and we choose the best number of Gaussians for the fit based on the Akaki information criteria. That's pretty common, actually. So we validated this in vitro with an accuracy of less than 10%, and I actually put some data in for this to show you what I mean. So for this sort of validation experiment, we used breast cancer cell lines. This is a HER2-positive breast cancer cell line. This is a triple-negative breast cancer cell line. And we co-cultured them. So we knew how many of each cell line we had in the dish, and then we did our imaging, and we compared to what we already knew. So the data here is in the blue rectangles. And then this red dash stuff, this is the 2 independent Gaussians that were fit. And then the blue here is the addition of the 2 Gaussians. So the errors that I plotted here, this should have a little line over it. This is an error in the mean, and this is the error in the proportion for the 2 populations. And I've written these here as percents just to give you an idea of how close we are to the actual proportion in the dish. So this is where I got that 10% number from. And I plotted this here just for the NADH lifetime, but we did it for all of our variables. So what this allows us to do is threshold our images just based on the lifetime so that we can find where the HER2-positive cells are and where the triple-negative cells are. So this was just a validation experiment, and it allowed us to define some parameters over which we knew we could identify subpopulations. So we took this data and some of our human tissue data, and we used it to do a simulation as well. And this is how we determined the minimum number of cells that we needed in our sample in order to identify a certain percentage of a subpopulation. So say we wanted to find a 10% subpopulation of cells, we needed 500 cells in order to do that. And this is how we define that. Okay. So today I told you it's going to be a heterogeneous talk, and so I had to make an outline to help keep myself on track. So first I'm going to talk about what we've done in animals, and this is where we take tissue from xenografts or spontaneous tumor models, and we grow them in 3D, and we compare the outcome of the mouse to what happened in the culture to see if we can predict what happens to the mouse based on what happens in the culture. So we have some preliminary data in pancreas and then some more data in breast, so I wanted to show you both of these. And then we have done the same thing in patients where we get a patient sample and we'll grow organoids, and we're waiting now for validation of this data. So we have only 1 pancreas cancer patient so far and a few more breast cancer patients. And then at the end, I want to talk to you a bit about how we're trying to quantify the heterogeneity of these samples that we get using metrics that we developed. And this is in an in vivo head and neck squamous cell carcinoma model. So like I said, heterogeneous, 3 different cancers here. And then at the end, I think we're making some progress in sorting cells based on their autofluorescence, which will be really helpful for us in identifying the features of those resistant cells that we identified. Okay, so first, I'm just going to talk about the animal studies. So is someone keeping track of time? Because I have no idea. Will you like wave at me when I run out of time or something? There's no clock in here. And there's no clock on here either, so hopefully I'm doing okay. Okay, so the question we're asking here is whether optical metabolic imaging, which is just fluorescence imaging of NADH and FAD, in these primary 3D organoids, when I say primary, that means that these cells never touch plastic. So we just take them out of the animal and grow them in 3D, a 3D matrix. We want to know if these can predict in vivo response, resistance, and recurrence in a mouse model. So I've told you a little bit about these organoids, but I thought I'd give you a little bit more background. This is what they look like, for example, in the pancreas. They can grow very big, actually, from the pancreas samples that we get. The breast samples, they seem to grow a little smaller. So anyways, these include all of the tumor cells as well as any stromal components that we get in the tissue. So we don't exclude supporting cells. We include all the cells in these sample. And we grow these in 3D conditions so that we can maintain cell-cell signaling and those 3D interactions that are important for maintaining the behavior of the tumor. And we want to use these so that we can measure cellular level response to treatment using primary tissues, which is relevant. So what we've done here in this PDAC model, PDAC is pancreatic ductal adenocarcinoma. These come from a knockout model, so these are very aggressive tumors, and they actually mimic human pancreatic ductal adenocarcinoma histology very well. That's why we chose this model. They're very fibrillar, so they have a lot of collagen in them. What we do is we take these tumors from the mouse, and we digest them and embed them in a 3D matrix. And then in this study, we imaged over 7 days. So we imaged 1, 2, 3, 5, and 7 days after embedding them into the 3D matrix. And at the time we embed them, we also treat them with drugs. So the drugs we chose are gemcitabine, which is really the standard chemotherapy for pancreas cancer. So most people get this. AZD1480 is an anti-JAK and STAT inhibitor. And as you'll see, this is something that is useful for improving drug penetration in pancreas tumors and their combination. So for validation, we actually took these mice and treated them with the same things that we were treating in the 3D culture, and we waited much longer for this. So this endpoint came, weeks and weeks later. We let the mice live as long as they could. This is, again, very aggressive, so they only live for about 58 days. And so we have endpoints of tumor volume, tumor weight, immunohistochemistry markers, and survival. I think I'm only showing a few of those today, though. Okay. So what was interesting with the PDAC model is that we got very heterogeneous organoids growing in our 3D culture. So we have these, we labeled type 1 and type 2, and then as I said, they're very collagenous. There's lots of collagen. So we had a lot of fibroblasts in our culture as well. So if we just look at these individually and look at their OMI index, they're quite different, especially between type 1 and 2 organoids. They have very different basal metabolic rates. So this is not with any drug treatment. And then the fibroblasts fell somewhere in the middle. So what we know about the morphology of these organoids is that they match the morphology from other studies. So other studies looked at primary PDAC in mouse models and also metastatic lesions derived from primary PDAC in mouse models. So these type 2 organoids look a lot like what would grow from the metastatic lesions, and these type 1 organoids look a lot like what grew from the primary tumor. Okay. Oh, and I just want to show this data because it was really exciting. These are very well separated. So these are the actual histograms for the data, where you can see the type 1 organoids, the fibroblasts, and then the type 2 organoids. So they're quite different in their redox ratio. Okay, so this is just one of the survival measurements I told you about. This is the tumor volume. So what we have here is the single agent treatments, gemcitabine and this JAK inhibitor, and then their combination. So I will tell you that different survival data spanned out differently. So this is a tumor volume, but if you look at tumor weight, only the combination treatment achieved any smaller tumor weight than the control. And in survival, just the combination treatment also won out over the other monotherapies. So again, it depends on what you look at, but this is generally what's going on with the tumors. So this was all done in vivo. These are endpoint measurements at the time that the mouse died. And so in a separate cohort of animals, we took out the tumor before they died, and we grew them in 3D culture in dishes like this. And first we looked at the OMI index in type 1 organoids. You can see here that they develop a resistance over time to this JAK inhibitor. And their response increases over time to the single agent gemcitabine. And then they have this sustained, very good response to the combination treatment. This is an agreement with standard measures. So after we finished, or no, this was a separate cohort. So this is at day 3. We did immunohistochemistry in these organoids, and we looked at apoptosis. So that's what cleaved caspase 3 measures, is apoptosis. And we see at day 3 increased apoptosis in the gemcitabine and combination treatment group, which is in pretty good agreement with what we see in the organoids. And again, I thought it would be important to look at the heterogeneity of this response. So this is looking at just the JAK inhibitor, so the one that developed a resistance over time. And you can see here's the control for comparison. So at day 1, there's a bimodal response here. And then by day 3 in blue, and day 7 in green, this response becomes unimodal and it completely overlaps with control. So this is sort of what I was talking about before, where you have maybe multiple subpopulations of cells that then mutate or die and leave a different heterogeneity. So in this case, it left a more homogeneous tumor. So perhaps this responsive cell population died. If we look at the type 2 organoids, they actually have a little bit different response over time. So this is why I think it was important to differentiate these by their morphology. So over time, these type 2 organoids showed a sustained response to the JAK inhibitor. And gemcitabine sort of fluctuated over time. And then again, the winner, I think, especially by day 7, was a combination treatment. So again, on day 3, we compared what we got to what standard measure is, which is cleaved caspase 3, a measure of apoptosis. And we see, again, gemcitabine and the combination treatment had increased apoptosis compared to control. So from this, it looks like we can somewhat predict what will happen in the animal based on single-cell analysis of these 3D cultures. So that's encouraging. And I think, as you know, I've mentioned a few times, pancreas cancers are very fibrous, and it's hard to get drugs to penetrate into those tumors. So this is only half the story. This is asking the question, if we can get the drug to the tumor, how will the cells respond? So the other half of the question is, can we get the drug to the tumor? And that's what we were trying to answer with this second set of experiments. So these are all tissue sections taken from the mice at the end of treatment. So what we did here is break them up into vehicle, single agent, gemcitabine, and the JAK inhibitor, and then the combination treatment. And these measurements here are imaging mass spectrometry, so they're specifically looking for the mass spec signal from this drug and gemcitabine. And so what we see is expected in the control, no drug gets into the tumor at all. In the AZD1480 treatment, we do see drug penetration. With gemcitabine, the drug's not getting in at all. So this is actually what we see clinically as well. This is in the mouse model. So this is a big problem. But then if we use a combination treatment of the JAK inhibitor and gemcitabine, we see that this drug is getting in as well as the chemotherapy. So that's important because it means if we use this combination treatment, we can get the chemotherapy into the tumor and achieve the dose that we need to kill the cells. So that's good. But then why is this happening? So from these same animals, we had different tissue sections, and we did SHG microscopy on these sections, and we quantified many things. I'm only showing this here. We quantified the collagen density and the alignment. So what I'm showing here is the alignment, and we see that the alignment is significantly decreased for the combination treatment compared to control. And I'm not showing you all the data. We also did trichrome staining and staining for collagen 1. And also, all of those measurements showed no change in the density of collagen. So all we're seeing here is a decrease in the alignment, but we see no change in the density. And this is important because people have shown, who know pancreas cancer, that if the density of the collagen matrix diminishes, if it becomes a softer tumor, those tumors then metastasize and become more aggressive. So we don't want to diminish the collagen matrix, but we want to reorganize it so that we can improve drug penetration into the tumor. And that's what we're achieving here. So that's good. So this was all done, again, in tissue sections taken directly from the animal after it was euthanized. So in a separate cohort, again, we had tissue samples that we grew in 3D organoids, and we looked specifically here at the fibroblasts. So this is a drug-responsive fibroblast to gemcitabine, the JAK inhibitor, and then the combination. So what you see here is kind of interesting. These fibroblasts don't seem to be affected very much by gemcitabine, the chemotherapy, but they are fairly sustained effects from the JAK inhibitor and from the combination treatment. So if we look at Ki67 here, this is a measure of proliferation. We see decreased proliferation in the AZD1480 and combination treatment groups. Interestingly, we saw no apoptosis with this treatment. So what we're doing with this treatment to the fibroblasts is decreasing their proliferation, especially with the JAK inhibitor, but we're not killing the fibroblasts. So in that way, I hypothesize, I'm not sure, but I think that this is why we're not seeing changes in collagen density, but we're seeing changes in the organization of the collagen. Okay, and then this was also a heterogeneous response over time. So if we look at this, control is in black, and then the red at day 1, day 5 is in green, and day 7 is in blue. You can see that the response becomes much less overlapping with control over time. And this sort of agrees with what we see over the time course of treatment. So from this, we think it's interesting that we can have some insight into drug delivery looking at tumors in a dish. If we look at the right thing, if we look at things like fibroblasts, and we know that those have an effect on collagen organization and density, we can start to tune what drugs might be helpful in delivering treatments. I need a drink of water. How much time do I have? Sweet, good. I have a lot more to say. Oh, I forgot. I need to transition. So I'm totally transitioning now to breast cancer. So those studies were all done in a knockout model of pancreas cancer, which is great because those tumors have an immune system and they have all those great things that we like in knockout models. But this one is great in a different way. We're using human tumor cells, and these cells had developed a resistance to the drug trastuzumab. Trastuzumab is a HER2 inhibitor that is clinically used, and it has an issue in the clinic as well in the development of resistance over time. So we thought this was a very clinically relevant model. This experiment is very similar to the one I just showed you in the pancreas. We took the tumors out and grew them in organoids and embedded them with clinically relevant drugs, and we imaged them at 1, 2, and 3 days after embedding them. So first we did what we did in the pancreas as well. We had a gold standard measurement. So this is the tumor volume over time. So here we have all these single agent treatments and then 3 combination treatments. So what I think is interesting here is, well, the trastuzumab treated tumors continue to grow as you would think they would because they are resistant to this drug. And then these 2 blue ones I think are interesting. Paclitaxel and XL147 initially show a response to treatment and then regrow over time. And then all of these combination treatments show sustained response over time. So this gives us a nice distribution of response data to compare to our measurements in a dish. So that's what we did next. We took a separate cohort and we took their tumors and put them in a dish and measured response. So what we have here is measurements at 24 hours and 72 hours post-treatment. And you can see initially that the paclitaxel and XL147 treated groups respond, and then by 72 hours they have regressed, just based on our single cell measurements. And then these combination treatments show a sustained response over time. And again, this is consistent with what we measure with standard measures. This is a measure of apoptosis at day 3 from the culture. So we see increased apoptosis in these combination treatment groups, as you would expect based on the metabolic data. So again, I think it's important to measure single cell response. So we wanted to see how this varied over time. So here we have the control and the trastuzumab, which completely overlap as expected because these are tumors that are resistant to this drug trastuzumab. If we look at paclitaxel, this is one that showed initial response and then regression. We see, again, 2 subpopulations of response at day 1 that then diminish into 1 population by 72 hours. And then the combination treatment, which shows 2 subpopulations of response, which then merge into almost 1 subpopulation that's very responsive and not overlapping as much with control. So from this, I think we can conclude that these measurements could be useful for increasing the throughput of drug response and resistance in animal models. So instead of measuring response on a single animal level, we could maybe measure it on a single dish level, and therefore we could look at more combination treatments, and we can look at dynamic changes in cellular response over time if we can access these in a dish. Okay. So that was the animal story. So now we've done a little bit of this in patients. And so first I'm going to start with the pancreas, and then I'm going to talk about breast cancer. And we have more patients here, but both of these are undergoing right now active research. So I thought I'd show you this because these are the first patient sample we got from a pancreas cancer patient. So we took the tissue and we didn't dissociate it into organoids. We just looked at it to see what the architecture looked like. So what you're looking at here is the NADH mean lifetime and the FAD mean lifetime at 2 different locations, location 1, location 2. So what we see here is an intraductal tumor, and you can see here there's lots of stroma, lots of cellular debris, and lots of heterogeneity just within the tissue itself. We see this here as well. So when we took these tumors and we dissociated them into organoids in 3D culture, we saw a range of morphologies as expected. So this one here looks like it might even be a duct forming from maybe a functional part of the pancreas that was embedded in the 3D culture. And then things look very different. So it was very, very heterogeneous, as we expected. So for this first sample, actually, we had some pathology ink on the sample. So we only imaged up to 24 hours because then the ink got all over our culture and we couldn't image any longer. So that just shows you, the first time you do something, something's going to go wrong, and you learn something, so that's good. But at least at 24 hours, we were able to image, and we saw a response to gemcitabine, and this again is our JAK inhibitor from the mouse studies, the combination treatment. So this is just in one patient, but this helped us learn about acquisition. It helped us learn about the heterogeneity of the tumor, what would grow in the culture, and if we could, in fact, measure drug response at all. So this was an interesting pilot study. So it just told us that we were able to really grow these tumors. So in breast cancer, you may know, there are many different subtypes of breast cancer. And so we looked at these 3 receptor statuses, triple-negative breast cancer, ER-positive breast cancer, and HER2-positive breast cancer. So here I've shown 1, 2, 3, 4, 5, 6 patients, but now we have more, but we're waiting on follow-up data from them. So what this showed us, actually in this initial study, we had 100% take rate in all of our studies, from the animals to the patients. We're always able to grow the organoids from the tumor, as long as there's some tumor tissue in the tissue sample that we get. So this is in contrast to, say, those patient-derived xenografts. I have a collaborator who actually gets the same patients that I do, and she can only grow tumors in these patient-derived xenografts from a subset of these triple-negative patients have aggressive tumors. So this is advantageous. If we just look at our first patient, this was an estrogen receptor positive patient. So they don't have a HER2 positive tumor, so they would not respond to trastuzumab. We measured response to multiple different drugs. I'm not going to go through all of them, at 72 hours. This is just to show that what we measured with metabolic imaging is in agreement with what we measure with immunohistochemistry. So again, this is apoptosis. We see an increase in apoptosis in all groups except for the trastuzumab group, which is in agreement with what we see with our metabolic imaging. And then if we look at changes over time, so this is at 24 hours in the same patient, 72 hours, which I showed you previously, you can see that there's a heterogeneity in this response as well. So this is the histogram corresponding to 24 hours, and this is a histogram corresponding to 72 hours. So if you just look, for example, at this 3-drug combination, the heterogeneity in this response is reflected dynamically. So here we have a very strong subpopulation here, and a weak one here, and here they're starting to balance each other out. So again, this was more of a pilot study, so we wanted to see if we could resolve single-cell response and if we could relate that to standard measures in immunohistochemistry. So I think this is promising, the fact that we can grow these and we can measure drug response. It means that maybe this could be used as a clinical screen to personalize treatment. So that's what we're working on right now in the breast. We get actually core biopsies from patients undergoing neoadjuvant chemotherapy. So they get these core biopsies at the time of their port placement, so before they've had any drug treatment. And we will grow multiple organoids from these biopsies and do the same tests that I've been showing you today. And what we're doing right now is waiting for them to undergo their chemotherapy and have their surgery so we can have an endpoint of comparison to see if our predictions here from these 3D cultures are in agreement with what actually happens to the patient at the time of surgery. All right. So at the end here, I just thought I'd talk to you a little bit about our efforts in trying to actually quantify the heterogeneity of this response and relate that to treatment outcome. And then a little bit more about autofluorescent cell sorting. So the question we're asking here is whether we can create a quantitative in vivo measure of tumor heterogeneity on a cellular level that will predict drug response. So there is this Shannon Diversity Index that we found, and it is a measure of heterogeneity. It's actually a population measure. So we've just modified this slightly to measure the heterogeneity of our histograms that we've been measuring. So all we did here was add a term. So what we're measuring here is the, where'd it go? The distance between the median of the population and the median of the whole sample, which is right here, and the median of the population, and then the proportion of these two populations, which is the p. So this heterogeneity index, that we've defined it here, it indicates increased number of subpopulations. If the heterogeneity increases, then there's more subpopulations. Increased equality in the weights of the subpopulations. So if these two become equivalent, it is a more heterogeneous population than if this is a small, say, 5% population. and increased separation of the locations of the subpopulations relative to the mean of the whole thing. So this is just one quantitative measure that we have to actually put into some sort of scalar value how these distributions look. So we validated this in a head and neck squamous cell carcinoma model. So I'm changing, again, the cancer that I'm talking about. So these guys are actually treated with cisplatin, which is a chemotherapy, and they're also treated with cetuximab, which is an EGFR antibody, because these tumors overexpress EGFR. So if we look at the response data, we can see that, I think this is dead, that the monotherapy treatments are sort of reducing this to what do you call it? Like sustained response, stable disease, that's what it's called. So the tumor isn't really growing or shrinking by the end of the experiment. But with a combination treatment, we see a general decrease in the tumor size. So this was in the in vivo validation study. In a separate cohort, we imaged in vivo at 48 hours post-treatment with all of these therapies on our microscope. So these, I think these are the first in vivo data I'm showing. So for this, these were flank tumors. We just cut the skin out and then imaged on the flank. And these are an example of what the in vivo images look like. This is the NADH mean lifetime. And this is what our histograms look like. So I've shown you these before. Like here's our control, here's the monotherapies, and here's the combination treatment. But what's different here is that we've actually quantified how heterogeneous these distributions, density models are. So the H index here is small for the control because it has only one population. And it's small here for the combination treatment because these two subpopulations are very close to each other. But for the monotherapies, you can see two very distinct subpopulations that are almost proportional to each other. So they have very high H index right here and right here. So if we look at this data and we compare it to the response data measured by tumor volume, we can see that the most homogeneous response is in the combination treatment, and it had the greatest response over time. And then the most heterogeneous response resulted in stable disease. So if you think about this and you maybe want to interpret it a little bit, the way I think about this is that this is two very distinct subpopulations of cells, some of which are dying and some of which are proliferating, and that just results in a tumor that doesn't change in size. So this is just for one animal model, and this was just our first attempt at this, but I think this heterogeneity index could be a useful measure of heterogeneous response in the tumor. So that measurement was just using one variable. So the heterogeneity index is applied to single-dimensional data, single histograms. And I told you before that we measure 7 variables. We're measuring the intensity and the fluorescence lifetime of each of those molecules, and that results in these 7 variables. So what we need to use is a data reduction technique. Most people use principal component analysis. What I'm going to talk about for this next 2 slides, I guess, is this VISNI. So VISNI is visualization of stochastic neighbor embedding, and we chose to use this rather than PCA because it doesn't require a linear transformation, so it maintains this multi-dimensional relationship between the variables in 7-dimensional space, essentially. So what it does is it calculates, so there's 3 parameters shown here, we're measuring 7, right? So we're in 7-dimension space. We can calculate a high-dimensional similarity matrix and reduce this matrix to preserve the multidimensional relationships on 2D. So essentially we're taking the distance between different cells and maintaining that distance on a 2D projection. So this is handy because it can be used to identify low density subpopulations. So what we're looking for here is outliers. We want to find subpopulations that are outliers from the main population. And that's why this is sort of advantageous. So anyways, we used this data reduction technique on all 7 of our variables and then plotted them on a 2-dimensional matrix to see how they looked. So here the control is blue, and I know it's hard to see, but it's all right here in this sort of thing I'm circling. And then if you look at the combination treatment, it's in red, and it's all over here, very little overlap between the combination treatment and control. But then if you look at the monotherapy, cetuximab and cisplatin, they are very heterogeneous, and they have overlap between control and the combination treatment group. So this conclusion is very similar to what I had for the heterogeneity index, but we have to use this kind of data reduction approach if we want to look at heterogeneity across all of our variables. And so we found this to be pretty useful. Okay. So I know I haven't talked much about our in vivo studies, and I think I just wanted to put this up here to acknowledge the fact that lots of the things we're measuring in a dish can't be replicated in a dish. Things like angiogenesis or collagen reorganization are very difficult to replicate in a dish. So we're also making these measurements on tumors in vivo in the same animal models. So we measure collagen organization with second harmonic microscopy, and we measure vascular density and oxygenation of microvessels using hyperspectral imaging. And we're relating these dynamic changes over time. So all of these things are dynamically changing, and they're obviously all related to each other. So it's important to look at these in different model systems. And finally, I haven't talked at all about immune cells. And immune cells have a really important role in response to therapy. So here, we've just started very preliminary studies. I actually didn't even know what a macrophage was, like probably 2 years ago, 3 years ago. And then I discovered they're really important things and we should be studying them. So macrophages in tumors exist in sort of a pro-tumor and anti-tumor phenotype. And so we're trying to replicate this in culture using known perturbations to see if we can metabolically discriminate those two populations of macrophages. And it turns out that lots of these immune cells, macrophages and T cells, actually use metabolism as a switch to change their phenotype. So this is true especially in T cells. They develop different metabolism to gain effector function. So we can be very sensitive to the behavior of these immune cells. And we're all doing all these studies in a dish right now, but eventually the goal is to use what we learn in culture and apply it to our in vivo imaging so that we can understand the immune response using our metabolic measurements. And I think I already talked about fibroblasts and their importance. Okay, so now the very last slide. So this is something, I think you always have to put something in that your student sent you like the night before. So this is that slide. So I think, you guys probably have this question on your mind. Okay, you find these subpopulations. What are they? What is the cell doing? What does a subpopulation mean? What are the mechanisms, essentially, for these subpopulations of cells that we're detecting? And I think that's a really hard question to answer, but I think if we want to make this measurement useful, we have to answer that question. So one way to do this is to do flow cytometry and flow sorting of cells, right? So if I can take the tumor and I image everything I want to image, and then I take the tumor out and dissociate it into single cells and do flow sorting, I can maybe sort those subpopulations of cells and study them more in depth to understand what are the mechanisms of resistance in these subpopulations. So that's the question I'm asking with this last slide. Can OMI subpopulations be sorted on a single cell level for further analysis? So we started again with cell lines that we understood very well. So this is again the triple negative cell line and the HER2 positive cell line. And then we mixed them up and tried to sort them on a flow cytometer. So the variable we're sorting with here is fluorescence at 405 nanometers using unstained cells. So we're trying to measure NADH intensity using a flow cytometer. And we're trying to sort cells based on their NADH intensity. And based on this initial study, I think we might be doing that. So this is what we get from the sorted population of 231 cells and the sorted population of SKBR3 cells. So then once we sorted these cells based on their NADH intensity in a flow sorter, we let them grow for 6 days, and then we image them on our 2-photon microscope. And we see the morphologies that we expect from 231 cells and SKBR3 cells. And we also see the expected increase in NADH intensity and SKBR3 cells that we have previously characterized. So from this initial experiment, I think it may be possible to sort cells based on their NADH intensity and then understand what causes these changes in our metabolic measurements. Okay. So today I talked about lots of different things. Hopefully you kept up. But what I wanted to tell you mostly was that imaging on a single cell level is important. I think understanding tumor heterogeneity is important. And I think if we can really wrap our heads around what's happening in the sample and understand how to replicate what's happening in vivo, we can use this system to streamline drug development and to inform personalized treatment decisions. So all of this work was done at Vanderbilt with my wonderful students and wonderful collaborators. I have actually all of these people worked on this data because I talked about so much today. So there's not a whole lot of people to single out, but Alex Walsh really did the lion's share of this work, and she's a postdoc now at the Air Force National Research Lab, doing wonderful things, as always. So, thanks. Yep.
People have been growing biopsies for years with colony assay and things like that, and it really hasn't paid off. Now, how is this any different?
Yeah, we ask ourselves that all the time. when I first started this experiment, I thought there was no way these results would come together. So I think there's actually more than just oxygen to worry about. There's glucose to worry about. There's oxygen. There's metabolic products like lactate. So the pH of the tumor is going to change over time. The substrate levels, everything's changing. The food source. And they dynamically change over time. So one way that I think about this is that I have a tumor sample and I have a control. So I have grown whatever piece of tumor I happen to get in a set of conditions, and I have treated it with no drugs. And how do the drug-treated groups compare to that control? So I can tell you for sure that the metabolism of the cells I am growing in my 3D matrix is not the same as what I measure in vivo, because we've done those experiments. We'll measure the metabolism, take the tumor out, dissociate it and grow it in 3D, and measure the metabolism. Is different because, as you said, the oxygen's different, the glucose is different, everything's different. So I think we might be getting away with it because we're comparing to a control. So having said that, I think there are many ways to control these parameters. People are making these bioreactors that are very well-controlled oxygen levels. You can have microfluidic devices with well-controlled flow of substrates and outflow of products. The matrix can be perhaps made to replicate what's in the patient based on radiographic measurements. So for example, in breast cancer, you can measure the mammogram and look at how dense the breast looks and use that to estimate what your collagen gel should look like. So I think all of these things are tweaks that are important, and we're thinking about them. It's a complicated question. And he's not going to ask any more. Okay. That I didn't talk about at all? Yeah. Well, what I thought was interesting, I didn't show the in vivo data, but when we measured like the redox ratio over time in breast tumors treated with trastuzumab, we saw the control was varying over time, and we saw that the treated group was sort of continuing to decrease its metabolism over time. And we thought, well, gee, why is the control changing over time? It must be due to the vasculature. Let's measure the vasculature. So we did, and it was changing differently over time. You know, so then we sort of got more questions to ask than answered. And we also measured the collagen, which interestingly, I thought this was really weird. In tumors that were not responsive to trastuzumab, so they grew anyways when we gave them trastuzumab, they reorganized their collagen architecture, even though the tumor was not responding to treatment. So, yeah.
Can you say like what spatial scale the reorganization happens? Is it at the sub-millimeter scale?
Of the collagen or the?
That reorganization.
Oh.
Could you observe it with a mesoscopic optical?
Oh, I don't know because I didn't try. I can look at the data, but I know that we image with like 2-photon resolution, and that's the resolution we were using. I don't know. Ask me on an email, and I can look at the data.
How much? So when you're imaging with 2-photon, you know, when you start getting slightly bigger tumors, how much does sampling error come into play? Because you're already talking about a heterogeneous
Yeah.
population. So if you look at multiple areas, how different? I assume they're pretty different. So how do you account for that?
We worry about that even on the biopsy level, right? So we'll get a biopsy from a patient or two. Actually, we get up to 4. And we think, well, did we catch a necrotic area or not? Did we catch a hypoxic area or not? I mean, it's, yeah. So there's sampling error across the entire experiment. For the xenografts, so I think it depends on the model you're looking at. The xenografts are pretty homogeneous, but as you saw, the knockout models, they're more heterogeneous. And we just hope we capture enough of the tissue to capture the heterogeneity present. Yeah. We're looking at single cells. We want to look at all the cells. Eventually, I want to look at all the cells in the whole tumor. Then that problem would be solved. Yeah.
You can do that with brute force.
Yeah, you can. I think I said that in the grant to make a reviewer happy, and I still haven't done it. No, I said I would grow the organoids from the entire tumor and characterize the entire heterogeneity of the tumor, and then quantify the number of samples I need to account for that whole tumor heterogeneity. Yeah.
So it's kind of along those lines, your colleagues who are kind of coming from the cancer center and more traditional cancer biology are thinking of tumor heterogeneity and genomic diversity in a different way.
Yeah.
About it. We're kind of looking at a very distilled downstream indicator. Do you think you'll be able to make the connection between
genes?
what most people are doing in cancer research?
Yeah.
Like 10 of us are doing?
Yeah. You asked me this question before, and I never did anything about it. I think you're right, though. Yeah, because everybody at the Cancer Center talks about genomic heterogeneity. And
so, Henry asked you about the heterogeneity of oxygen and nutrient distribution, but you're capturing the genomic diversity directly from the patient.
oh, yeah.
And so maybe that might not be all that important for you to get a snapshot of the heterogeneity and metabolic diversity of that tumor. Initially,
Maybe.
just to get some insight.
Yeah.
But do you think that like the OMI or PD, how could you come up with something that's actionable for a clinician? Everybody wants to have some understanding of the diversity of the tumor. And how are you related to the genomic diversity?
I think,
Could you do that?
I don't know. That's a great question. I think lots of our patients get like genetic screens, and I know the place to start would be to look at that and compare it to what we get, like the heterogeneity of the tumor versus the heterogeneity of the genome. That's like the 0th order answer, right? But then actually crossing the bridge from the genes all the way up to the metabolism, that sounds like a fun project to work on. But I think there's a lot of work to be done, yeah. Yeah, go ahead, you start it. No, it's actually a really good point, yeah.
Well, maybe because the drug targets are either molecular or metabolic, you have a better chance of having some interpretability of the metabolic diversity that could be directly actionable.
That's true.
Not much is actionable with genomic diversity.
Yeah, that's true. The drugs we choose target specific pathways that are determined by the genes of those cells. And we always use a PI3 kinase inhibitor as like a positive control, negative control, because it really just shuts down the metabolism. So that's like a metabolic measure, right?
Yeah.
I guess if you get enough throughput, you can measure tons of different targets, and that's how you would relate them, right? Sounds fun. Mm-hmm.
It's shown that the average lifetime of either in an age or a they don't really relate to the. But what about the, because it's a bifunctional DNA, it's a bi-exponential DNA. So what about the ratio of the pre-exponential factors or something? What about that? Does that relate? Because that's what sort of
Oh, you're wondering, I see. You're wondering because I keep showing the mean lifetime, if the individual lifetimes correlate more.
Or the alpha 1 and the alpha 2.
Yeah, they're all, actually we put this in like a supplemental figure. There's this big table where we describe the behavior of tau M, tau 1, tau 2, and alpha 1. And they're all behaving differently. So I think, again, they're somewhat correlated, but they're each measuring something a little bit different. So I'll tell you what I think, though. So I think that alpha 1 is the one that people like to talk about because it tells them the relative amounts of free and protein-bound molecule. And that's easy to interpret, and you can explain it to people, and they know what you're talking about, right? But we see changes in like the lifetime itself of like the protein-bound lifetime. And we think that's due to differences in the protein binding activity of NADH. So when it binds to lactate dehydrogenase, it has a different lifetime than when it binds to pyruvate dehydrogenase. Yeah, so we think that the lifetime itself changes depending on the preferred binding sites. So, and that's different than how much of it is bound. Yeah. So all of it's useful. That's why we need multi-dimensional data reduction. Yeah.
Well, to put you on the spot, because we have a lot of LFT people here.
What's LFT?
Laboratory fluorescence dynamics.
Oh, okay, yeah.
So, can you compare the approach to multi-exponential decay and trying to interpret the, fractional contribution as a relative to the phaser approach? And are you just not using phaser because we haven't converted you?
I told you, like, when I met Enrico Graton, who was I telling this story to? You. someone today. The first time I ever met him, he goes, "Give me your data." I was like, "Who is this man?" Italian accent, shady. But Peter Shaw was right next to him, and he said, "You can trust him." I said, "Okay." We do actually use the phaser approach. We use it in all of our studies as a sanity check because we're really nervous about fitting data. As I think that's why people invented this phaser approach, right? But for interpreting the data, we usually show this in our papers because we have an easier time explaining it and interpreting it in terms of physical things like alpha 1, tau 1, tau 2. But we always plot the phaser just to make sure we're measuring NADH and it's a 2 exponential decay and all those things you want to know. Phasers are great.
Okay, that's everything then. Let's thank Melissa again.
All right.